Welcome to the deep dive. Today we're diving into something that, well, sounds incredibly simple on the surface, just taking a picture of living cells.
Right. Sounds easy enough, like using your phone.
Exactly. But the reality, it's vastly more complex, often really frustrating. We're talking about capturing something almost invisible, constantly moving inside this perfectly controlled, tiny world.
And not just for a second, but for days, maybe even weeks. It's high-stakes stuff.
Absolutely. So our deep dive today is pulling back that curtain on live cell imaging. We've drawn insights from dozens of conversations with scientists, researchers, experts right at the forefront. Our mission here is to uncover those surprising, sometimes baffling daily hurdles these folks face, the ones pushing the boundaries of biology. And crucially, we want to pinpoint where innovation is really, really needed.
Yeah, you're going to hear about the grit, the clever workarounds, and honestly, just the sheer persistence it takes. And what's really striking, I think, is the contrast. You have this incredibly cutting-edge science, gene editing, bioprinted tissues, tiny human organs growing in dishes. It's amazing stuff.
It sounds like science fiction sometimes.
It does. But then the actual tools, the infrastructure, the day-to-day workflows for seeing these things, they often lag way behind.
So it creates these hidden roadblocks.
Exactly. Hidden taxes on progress, you could say, loads of frustration. It really slows things down. And that's what we want to shed light on for you, those real human challenges.
Okay, let's unpack this. First big area, that incredibly delicate dance of keeping live cells, well, happy. Environmental control and stability.
Yeah, it's not just about keeping them alive, is it?
No, exactly.
Yeah.
They need to be comfortable enough to behave naturally. Otherwise, your whole experiment could be invalid. So what's tripping scientists up right at this basic stage?
Well, this is where seemingly simple things become huge problems. Take humidity and CO2, critical stuff for mimicking the body.
Okay.
But many of these stage-top incubators, the little chambers right on the microscope, they actually struggle to keep those levels optimal. We heard from folks like Aditiran, Suchinbayan. There's this constant worry about CO2 levels dropping.
Really? So the cells aren't thriving?
Often, no. They do much better back in the big traditional lab incubators, so scientists end up moving them back and forth, which isn't ideal.
No, that sounds disruptive. And you mentioned condensation.
Oh, don't get me started. Condensation is a nightmare. Aditya described it vividly, this pervasive fogging on the heated glass lids, even after letting it settle for half an hour.
And that affects the image.
Totally. You get grainy pictures. It can, quote, screw up all your imaging. It's like trying to film through a steamy window just ruins the clarity.
Wow. What a perfect analogy. So it's not just the internal environment, but the equipment itself causing problems.
Precisely.
And these sensitivities, they don't just stay put when the cells are on the microscope, do they? What about moving them?
Ah, yes, transport. Even just moving samples between labs or, across campus, it's surprisingly risky.
How so?
Well, people like Jesse Gordon-Blake and Farzane Mirfakhar talked about worries over temperature swings, keeping things sterile, light sensitivity for certain samples, and of course spills, just dropping them.
Oh, no.
Yeah, it gets almost comical. Scientists invent their own little solutions, wrapping dishes in layers of parafilm and foil, just basic stuff to try and protect the samples during transit.
It really shows the hidden effort involved, doesn't it?
It really does. So much care.
So when you think about all that hidden labor, the lost data, just because of these basic environmental issues, it's like trying to build something amazing on shaky ground.
That's a good way to put it.
The key insight here maybe isn't just that cells are delicate. It's that these seemingly small environmental wobbles create this fundamental tax on science. Burns time, wastes expensive materials, and can undermine the data before you even start imaging.
Absolutely. It's a foundational problem.
Okay, so moving from the environment, let's talk about the actual imaging. Getting that clear, consistent picture of life happening in real time, especially over days or weeks. That's not easy either, is it?
Not at all. There are some major technical hurdles just trying to see clearly.
Like what? What is the biggest headache?
Well, one huge one is just keeping things in focus.
Yeah.
Cells move, especially over long experiments. We're talking 72, 96 hours, even 2 weeks sometimes. Arkita Sharma mentioned this.
They just drift around?
They migrate, they divide, they move out of the focal plane. So you're constantly losing focus.
Yeah.
And if you can't track individual cells consistently, that messes up your statistics big time.
Right. You lose the thread.
Exactly. And for thicker samples, like tissues that aren't stuck down, Yuxuan Huang pointed out you often need manual adjustments, constantly tweaking the focus, which is, incredibly tedious.
I can imagine. And then there's the light itself, right? Isn't that a problem?
Ah, yes. The big catch-22, phototoxicity and photobleaching. Mandy Wang explained this well. To see fluorescently labeled cells, you need to shine light on them, make them glow.
Okay.
But that very light can actually damage or even kill the cells. That's phototoxicity. Or it can just make the fluorescent signal fade over time. That's photobleaching, especially if you need high resolution, which requires more intense light.
So the act of looking can harm what you're looking at.
Precisely, and it gets worse. Even handy features like autofocus, which seem helpful, they can end up blasting the cells even more with the light, as Mandy put it, just zapping them repeatedly.
Wow. Okay, and what about those more complex structures like 3D tissues or organoids?
Yeah, that's another level of challenge. Imaging through thick 3D things like bioprinted constructs or these mini organs. It's tough. Amanda Jurowski, Derek Wells, Prabodha Purkayastha, Shreyas Gaikwad, they all mentioned issues.
Like what kind of issues?
Light scattering is a big one. The light just bounces around inside the tissue, gets distorted. It severely limits how deep you can actually see clearly. Prabodha mentioned that even with special long-distance lenses, you might only get clear images a few millimeters in.
So you can't see the whole picture.
Often, no, not easily.
It sounds like a constant fight against the physics of light. But it's not just the hardware causing frustration, is it? Yeah, I hear the software can be tricky too.
Oh, absolutely. That came up a lot. Users like Archita Sharma and Christine Ho described microscope software as, well, chaotic. Too many functions, options hidden away, just hard to learn and use efficiently.
Even for basic things.
Yeah, even simple stuff like adding a scale bar to an image could be a struggle. Farzan Mirfakar had this great analogy, comparing Zeiss software to using an iPhone, maybe more intuitive, simpler, but less customizable. Okay. And Nikon software, more like a Samsung phone, potentially more powerful, more freedom, but maybe a steeper learning curve, less immediately user-friendly. It just highlights that user experience frustration.
That makes sense. And weaving through all these specific challenges, there seems to be this fundamental trade-off scientists keep facing.
Yes, the constant battle between throughput and detail. Do you want to image lots of things quickly, like scanning a whole 96 well plate? Or do you need super high resolution on just a few cells to answer a specific biological question?
You often can't have both easily.
Exactly. It's a compromise. Hafiz Udin, Shreyas Gaikwad, Paige Kundev all touched on this. And it leads some people, like Hafiz Udin and Priya Makajani, to wish for, a smaller, maybe more affordable system, something that gives them really good, detailed data, even if it's not processing hundreds of samples at once.
Interesting. So stepping back, the big picture on imaging seems to be these constant, difficult compromises. Battling physics, battling clunky tools, sacrificing speed for detail, or vice versa.
Yeah, that sums it up well.
And this leads to another interesting point, that desire to maybe avoid fluorescence altogether.
Right. Christine Ho mentioned wanting non-fluorescent methods specifically to get around that phototoxicity problem. And you see people like Suruchi Poddar using simple, bright field imaging, just normal light for basic checks on cell health. Even though it doesn't give you as much specific information, it avoids blasting the cells with harmful light.
Okay, let's shift gears again. We've got the cells happy, we've managed to get some images. Now what? The data itself. This seems to create a whole new avalanche of problems. The data deluge.
Oh, absolutely. First, just the sheer amount of data is staggering.
How much are we talking?
Well, think about it. An experiment might generate gigabytes of image data every 20 minutes, and it could run for days. Aditya Ren, Christine Ho, Jodar Banata, they talked about ending up with these huge data sets. Terabytes, easily, from one long experiment. Videos, stacks of images.
Terabytes. Wow. So where does it all go? How do they store and move it?
Well, that's often surprisingly low tech. We heard from Anish Vasan, Christine Ho, Melissa Hernandez, Jesse Gordon-Blake. Manual copying. Using USB sticks, external hard drives. Still very common.
Seriously? USB drive?
Seriously. And as Melissa Hernandez pointed out, those drives fail. All that precious data, gone.
Oh, heartbreaking. What about the cloud? Isn't that an option?
It exists, sure, but it's often restricted. University IT departments can be wary security concerns, preferring their own in-house storage systems. Charles Keller, Connor Schlichting, Omar Kamal, Eunice Allen all mention these kinds of limitations.
So scientists want cloud solutions, but can't always use them.
Pretty much. They wish for easy cloud-based analysis and secure sharing. Christine Ho, Priya Makajani emphasized this, but the reality on the ground is often cumbersome. Imagine trying to manage thousands of HD videos on, like, an old laptop with a slow external drive. It's kind of the vibe sometimes.
And just keeping track of it all, organizing it must be a nightmare too.
Totally. Jesse Gordon Blake and Ann Anjum talked about how data often isn't organized systematically. Finding a specific image from an experiment run months ago, good luck.
Oof.
And linking that data back to the specific experiment conditions. Often manual, as Melissa Hernandez said, or maybe just a basic spreadsheet like in Samantha Seymour's lab. And if someone leaves the lab, Sujin Bayan mentioned how hard it can be to access or make sense of their past data.
It's like a library where the books are just piled up randomly.
Exactly. So much knowledge potentially lost or just hard to find. Huge waste of time just searching.
Okay, so storage and organization are messy. But what about actually analyzing the data?
Yeah.
Extracting the insights?
That's another major bottleneck. Processing these huge data sets needs serious computing power. Very powerful computers, as Christine Ho put it. Not always available to everyone.
Right.
And automating the analysis, things like counting cells, measuring their size or intensity. Many labs still struggle with this. They relied on really tedious manual methods, or they need complex custom scripts, which is a huge pain point for Aditya Ran and Jenny Urbina.
So it's slow manual work.
A lot of it is. Christine Ho called it donkey work. It just eats up time that could be spent on, actual scientific thinking.
And this is where AI and machine learning come in, presumably. Or try to.
Exactly. There's a lot of interest there. Using AI for tasks like cell segmentation, basically teaching the computer to automatically find and outline each cell in an image. Aaron Richards, Nicholas Francis, Per Boha, Per Kayasta mentioned this.
That sounds useful.
Hugely. Or even predicting how cells might behave, which Chia Ching, Josh Wu is looking into. But there's a but.
Okay.
Many biologists just don't have the coding skills or expertise to actually implement these AI tools effectively. That's a challenge Jenny Urbina and Nicholas Francis highlighted.
So the tools are there, but hard to use.
Sometimes, yeah. And there's also some skepticism. Ibrahim Syed and Vadim Jucode mentioned concerns about, quote, image manipulation, or whether journals will accept AI-analyzed data, or just the black box problem, the AI gets an answer, but you don't always know how it got there.
Right, the transparency issue.
Exactly.
So the core insight on data seems to be this digital chasm. Scientists are generating floods of data with advanced imaging, but they're often stuck with outdated or difficult tools for managing and analyzing it.
That's it. There's this disconnect. Computational experts want to help, biologists need the help, but bridging that gap is tough. Priya Makajani, Farzan Mirfakar pointed this out. It's a major bottleneck for discovery.
Okay, so let's say, hypothetically, the perfect incubator, the perfect microscope, the perfect analysis software exists, getting it into the lab. That's another hurdle entirely, isn't it? The practicalities, acquisition, budget, just finding out about new stuff.
Oh, yeah. Another battlefield. First off, cost. Lab equipment is incredibly expensive.
We're talking big numbers.
Definitely. Connor Slipting and Simona Hankova mentioned costs often blow past departmental capital budget lines. What might seem like nothing to a huge university could be a massive investment for a smaller one, or especially a startup, as Ibrahim Syed noted.
And funding is tight too, I imagine.
Always. Funding cuts from big agencies like NSF, NIH make it even harder. Morgan Opal brought that up. It's a constant pressure.
So, assuming they can find the money, actually buying it isn't straightforward either.
Not usually, no, especially in big organizations. Lauren Perez and Simona Hankova talked about navigating layers of management, bureaucracy, endless paperwork.
Sounds draining.
It is. Often, Simona Hankova mentioned, you need to show a common need, multiple labs or researchers needing the same equipment just to get approval. That takes coordination. And for startups, Mandy Wang highlighted how slow vendor responses can be crippling because they have that limited runway, finite cash.
Yeah.
Delays really hurt them.
Okay, so budget and bureaucracy are huge. But how do scientists even discover these new technologies in the first place? You mentioned it's not like scrolling Instagram for microscopes.
Haha, no, definitely not. Farzan Mirfakar confirms social media isn't really a primary source there. It's much more traditional, really.
Like what's...
Reading scientific papers, talking to colleagues, peer recommendations are huge. Attending seminars, workshops, conferences. That's what Anam Anjum, Arkita Sharma, Farzan, Hafizuddin, Mandy Wang, Terry Ching all pointed to.
So networking and staying current with the literature.
Exactly. And direct interaction with vendors is key too. Getting demos, seeing the equipment in action. Mandy Wang stressed how important that is for making a decision. It's very much built on trust and seeing proof.
And I guess that leads to a preference for the familiar. sticking with what you know.
It often does. Scientists tend to gravitate towards the big established vendors, Nikon, Zeiss, and specific models they've used successfully before. Mandy Wang, Chi Ching, Josh Wu, Farzan Mirfakhar mentioned this.
Makes sense. Less risk.
Yeah, you're investing a lot of time, money, and experimental hope, so you stick with trusted names. But you do have to wonder, like you said, does that loyalty sometimes make it harder for genuinely disruptive innovations from smaller players to break through?
It's a really important question. So the insight here seems to be that even when great solutions might exist, the road to actually getting them into the lab is littered with financial hurdles, red tape, and relies heavily on traditional networks and brand trust.
That sums it up.
It really underscores the incredible resourcefulness of these scientists, doesn't it? Despite all these obstacles, the tricky environments, the imaging challenges, the data mountains, the purchasing hoops, they find ways.
They really do. We heard about people custom building parts, adapting old equipment, finding clever workarounds, Terry Ching, Hafam T, those Eman meeting discussions. It's a testament to their ingenuity.
So let's pull this all together. What's the big takeaway from this deep dive?
Well, I think it shows that live cell imaging is this amazing field full of dedication and brilliant problem solving.
Absolutely.
But it's also incredibly fundamentally ripe for innovation that addresses these daily grinding frustrations. We've heard about everything from fighting condensation and keeping cells in focus for weeks.
Right, to wrestling with terabytes of data and software that feels like it was designed in the 90s.
Exactly. These aren't trivial issues, they're major roadblocks.
And tackling these pain points, as you call them, could have a huge impact.
Massive. New technologies that solve these problems could unlock completely new insights into biology, speed up drug discovery significantly, and maybe just as importantly, let scientists actually focus on the science, not the donkey work, as Christine Ho so perfectly put it.
Free them up to think.
Precisely. Imagine the new questions they could ask, the new avenues they could explore if these foundational things were just easier.
It really makes you think. Given how incredibly resilient and clever these scientists already are, working around all these limitations, imagine the pace of discovery if those core challenges were truly solved.
The potential is enormous.
What if, like Christine Home used, taking videos of the basic building blocks of life became as simple, as ubiquitous as filming on your smartphone? What new worlds would open up then? What revolutionary insights are waiting just beyond these current frustrations? That's something to really ponder.